Bonjour, bienvenue à ce colloque de l'IHU Méditerranée Infection qui marque les 300 ans, un anniversaire de 300 ans de la peste à Marseille, et nous sommes réunis cet après-midi dans les
300th anniversary of the plague in Marseille.
Le Sud, et j'ai le plaisir, en le remerciant,
And I'm
d'accueillir le président de la
happy
région, le président Renaud Muselier, qui va introduire ce colloque cet après-midi.
I'm very sorry, I have a problem of echo on the translation. Throughout the Mediterranean basin, I have a problem of echo on the translation. When I speak, I can hear my voice. I'm very sorry.
Do you want me to take over? Shall I take over?
So in the Mediterranean basin, we've had all kinds of experiences, and the CHU has played a very important role. This morning, we had lots of rich discussions, and there were many speakers who have explained to me that...
So you received Professor Raoult, and with regard to the chloroquine, we received the General Director of the World Health Organization and the European Commission. And Professor Raoult was able to talk calmly, peacefully, with due respect, so that we can have a normal discussion, as we need to in a world where everybody respects each other. And I think that this is really what comes out. It's the respect of our populations, the respect of our differences, the respect of different opinions, so that we can add up the different competencies and find solutions. And this is what we're seeking. So thank you for this conference. Thank you, President. I do think indeed that respect of others is one of the characteristics of our South region. And this is something that we cultivate in the University Hospital Center with Didier Raoult. So we have a great number of students within different disciplines around infectious disease, and in particular students from the South who come from the southern shores of the Mediterranean and beyond. I am going to welcome the Professor Didier Raoult, who will be introducing this conference, which once again marks the 300th anniversary of the plague in Marseille. Sorry, I think we have a sound problem. We have a problem of sound. I'm very sorry. We cannot proceed with the translation. We cannot hear Professor Didier Raoult.
Non, ça ne fonctionne pas, Didier, pour l'instant. On ne t'entend pas.
Ça va lui plaire. Je pense que ça va lui plaire.
Non, ça ne doit pas lui plaire, mais...
I'm sure he's going to be very happy. Well, no, I don't think he will be very happy, but we're just going to wait for a few minutes whilst we're waiting to solve this technical problem.
Très bien. Voilà, ça fonctionne. Ça fonctionne. Très bien.
Donc, je vais faire une présentation pour expliquer la permanence d'un certain nombre de...
I would like to explain work within the scope of epidemics. So let's have the first slide, please.
Vous pouvez passer la première diapositive? Non, pas de diapositive.
Ça arrive.
Voilà, la deuxième, s'il vous plaît. Voilà, cette diapositive, une diapositive.
So this slide is a slide that you might see again, but we've just published the best review of infectious disease with a special edition on the plague. So what was interesting is how doctors dressed to protect themselves from the disease. And the family who does plays, and we have the rat, and fleas, etc. And people had trouble at the time dealing with the reality of the plague. And what is very interesting is a plague saint called Saint Roch. And Saint Roch comes from Montpellier. And he was born during the plague. He treated patients, and he's the Catholic saint patron of physicians and surgeons. He suffered himself from the plague, and for multiple reasons, he ended his life for a period of 5 years in Italy. But he is recognized as the patron saint who is mentioned every time we're faced with a pandemic. So there are a number of pests, a number of epidemics that faced, of course. Showed something. This is a painting from the Renaissance. He showed the bubon. That was a bubonic plague, and it affected the gland in the armpit. So you could clinically recognize the plague if the patient was observed. And with Michel Goncourt, we have noted something else. In Marseille, of course, there was the big plague of 1701 that killed a large part of the population. Therefore, one day, we wondered whether we could do a PCR diagnosis of the plague on collected when the tunnel was dug under the Charity Cathedral. And we hadn't planned this. We made the diagnosis by developing a new technique and using teeth, and we published this in the best journals. But there was a popular theory in the press that the plague was not due to the germ that we knew that was identified by the Swiss researcher, the Pasteur Institute, but that it was a hemorrhagic fever, therefore Ebola. Because we must say that what Camus said, if the plague comes back, we had a small episode of plague in Iran recently, and our collaborators worked on this. It's very easy today to treat it. We know what the treatment is. And the possibility of having a new plague epidemic in developed countries is very unlikely because it's transmitted by hectoparasites and the treatment is very simple. However, the risk of having an Ebola epidemic is something that is terrifying for the population. So he wrote a bestseller saying that the plague was not due to this microorganism. Yersinia pestis, but that and so it presented not only the sale of the bestsellers, but the demographic studies that to question the possibility that this was the plague in the Middle Ages. So this was the object of an incredibly violent debate, because another team, an English team who had no experience whatsoever in PCR and diagnosis, failed to reproduce our work. These are scientific problems that do occur sometimes. And this team had a great deal of influence over the major scientific journals, and they sent a quantity of information to Wikipedia, which meant that for a period of 15 years, our results were disputed by a certain number of scientists. Although the results were building up little by little, results from other teams to show that it was Yersinia pestis that we'd known since the beginning of the 20th century and that was behind the plague. And what is very interesting, so I'm afraid we're having connection problems. So there were there's information from the BBC that explains that.
La diapositive suivante. Oui. La grande peste et donc c'est sorti sur BBC, un grand documentaire.
So there was a documentary that was shown on the BBC, and they said no, it's not justice. So actually, all of this was a support to say no, they must be mistaken because the DNA is too deteriorated for us to identify it with PCR methods. So if they found it, it's because they were mistaken. So you can see the final conclusion of the BBC in 2002 was that it wasn't really the plague, it was something else. Because it wasn't a black plague. So there was a doctor from the Faculty of Medicine of Montpellier who did and he rudimentary knowledge ganglions. I'm very sorry, but he keeps cutting out. Next. Between the work that we did in 99 and the final article which says no, it is true, which was published in Nature, Nature published a draft genome of Yersinia gestis using the same technique as we did on teeth, and this was published in Nature. None of the 17th papers published on the same subject before, which showed exactly the same thing. So that shows that the scientific war doesn't date from today. It has always been this way, and either people didn't that you published or forget that you published it before you. So it's no child's game. There is no kindliness among researchers, no more than in other groups of the professional populations. So now we can come back to the Middle Ages. The plague was identified also by a German team and our team that revealed the presence of Yersinia pestis. And this study shows an analysis that was from strains that were 5,000 years old, and which no doubt explain the disappearance of a certain number of civilizations and villages that were affected by the plague, including in the north, where people who were hostile to the plague said it wasn't possible because there weren't the rats that we observe now and that came in the Middle Ages. So, based on a legend built around rats, fleas, and men solely, we have opposition to scientific hypotheses that date from another time, from people who don't really know what they're talking about, who haven't had the opportunity of observing and reading the existing scientific data, although this scientific data has been available for a certain amount of time. So you can see that the epidemics of the time raised the same type of problem. We're particularly aware in Marseille, as I was saying this morning, because it's a port. And the diseases from the south and the east came through Marseille, across the Mediterranean. Of course, the sea traffic explained the transfer of a certain number of infectious diseases. Infectious diseases to transport and to the war. That was for which there was a transport of troops. So things are easier maybe for other data, but scientific controversy is part of the picture. We think that financial aspects in Marseille, it's the financial influences which meant that we brought a lockdown, and the authority of the spoken word. was a problem at the time around contagion, because we were not familiar either with the microbe or the vectors of the disease. And therefore the question of contagion raised absolutely incredible controversies, as the same as for the causes. Some people believed it was a divine malediction. Like now, some people think it's a virus that was created in this or that laboratory. Therefore, society is brought together, and we see the incapacity of managing an unexpected event. Thank you very much, Didier Raoult. Thank you very much for this introduction, which clearly roots this conference in modernity. So we are talking about facts that occurred in Marseille and Provence several centuries ago now. But we can see that the different interventions are really anchored in modern day. And it's a chance that this conference on the plague is happening in the very year where Marseille, our country, and the whole world is faced with the COVID pandemic. So we can see the relation between both. So I suggest we continue the presentations, and we can now welcome Rémi Barbery, who is a doctor now, because he just defended his thesis a few days ago in the laboratory of the University Hospital Centre, and he's going to present the work that he did within this framework of our laboratory with Didier Raoult. And he illustrates the fact that all of these disciplinary scopes, paleomicrobiology, which is the fact of tracking with different methods the ancient microbes in human or animal remains, this thematic theme was born here in the Faculty of Medicine in the IHU Mediterranean Department of Didier Raoult, and it has involved dozens of research teams, mostly in Europe and other countries of the world. So it's something that was born here in Marseille in Provence, and Rémi Barbery will be showing you how this has changed in recent years, and will be presenting a specific aspect of his research. Rémi, it's up to you now to present your work. Thank you very much.
Merci beaucoup. Est-ce que vous m'entendez bien? Vous m'entendez? Oui?
Oui.
D'accord. Donc, première diapositive, s'il vous plaît. Donc, je tiens tout d'abord à remercier l'IHU, la région, pour cette invitation à ce colloque. J'ai le plaisir aujourd'hui de vous présenter les résultats d'une méthode que l'on a mis en place, une méthode interdisciplinaire entre deux laboratoires, le l'IHU Méditerranée Infection d'une part avec le professeur Michel Drancourt et le laboratoire ADES avec Michel Signoli. Donc c'est une nouvelle méthode d'analyse des textes historiques que nous avons essayé de développer pour pouvoir identifier de probables sources de peste.
This is a proof. The plague of Marseille of 1720. So next slide.
Donc, ces deux épidémies de peste ont lieu dans le cadre
These two pandemics took place. It began in 146 in Kaffa, came over the Black Sea, and people were starting to die from the plague on the ship. The people from Genoa fled, and the Black Plague invaded Europe. It lasted from 26, and there were 30 million victims. Then the plague invaded Europe, and there wasn't a single year until the 17th century that there wasn't an outbreak of plague. And I think later on this will be presented far better than I could. So what we can see is that there was a diversity of strains, and the plague had reservoirs, no doubt, in the northeast of Europe, and alternative hypotheses talk about several waves in Europe. So the big question, which has not yet been solved, is how was the plague transmitted? Was it the rats, fleas, or something else? Because the plague of the second wave was very different from the third wave. And there is an explanation. So there was work that was done in... And I'm afraid the sound is cutting off. And so it was in 1911, and there were transmission tests about transmission, and there was these were based on a mathematical model. So our goal was to reanalyze So we have documents on historical tests that document the plague. There was a plague for which we don't yet have immunological documentation. So we have texts from the 18th century, and it's the first time that we have possibility to analyze with a specific software to analyze the DNA. And of course, we have a sequence of letters, as you can see, and I will come back on a few results, striking results from this study. Firstly, we took the digital text from Google Books and we imported it into the software that identified the text that had words in the 18th century. And then everything else was deleted. And that gave us a list of words that were enriched with the control texts. Then we did a well club, and we have there are network presentations. So next slide, please. So first of all, the first thing I wanted to show you. So this is the results of the plague in Marseille. We have different indexes here. At the time, for example, there are 30 or 40 pages of texts where we do a deviation analysis to see if the texts are very different, or there are texts which are more similar. And so this gives us samples that we can compare with different microbiologies.
So here we have an example of some data processing in R. We've imported documents from over 1 million sources, and we've analyzed the words which corresponded to existing words in the French vocabulary, the 1.6 million words, and then we used words in a different way. And we wanted to find the words that were present at least 5 times in all. So here we have the criteria of selecting our statistics, which we used. So we asked for those words of interest were enriched, more words, and those words had to be more present in the test documents rather than the control documents. And so there was an adjustment of 0.05. Those are the words in red that you can see. And then we generated a word cloud, as you can see on the screen, 72 significant words in the text which we analyzed compared to the control text. And all of the words are used frequently in texts pertaining to plague. You can see mortality, contagion, perfume, plague, contagion, contagious, quarantine. The idea was to replace these words in a lexical context. So we used a PCR method and were aiming to define. We could ask the software to take a look at 15 words before and 15 words after, and determine what the significance was before and after. So it worked very well. For bubonic, for example, we noticed that it was in the armpit, the ear, the thighs, and then of course there was another word which was associated with that, which was coal that was applied to skin during the plague. So you can see it works very well for the word goods. The plague arrived in Marseille on a commercial ship that came from the east, carrying on board people who were infected. They also had goods which were infected. And I would like to take a little moment here to look at this slide here. This was a gesture which was supposed to be effective against plague back in the day. We analyzed all of this, and it transpired that, as you can see on this painting here, the bubonic pustule would be pierced. There would be an incision, and then the lymph node would be removed. The wound was cleaned, and that particular gesture seemed to be rather effective, and it seemed to bring down the mortality rate by over 10%. So that was used, that was something which we discovered. So to come back to our words, we categorized our words into different categories. So we place them in a category, for example, of words that describe the consequences of plague, or the nature of plague, or the public reaction to plague, etc. And obviously, we were very interested in the source of plague, how it got to Marseille. And there were a lot of words here that are involved. We wondered how many times these different words were used when we were talking about the source of the plague. So goods is at the top of the list, 69%, tatters, movables, things which you can move around, and clothes, of course. So the word air, of course, is there as well. That means two different things. It can be a source of plague, and it's also used to purify the plague, because it was thought that air could purify the goods that had been infected by plague. So there's a double meaning there. Words continue. There are more on the list there. The feathers, of course, but these are feathers in mattresses, for example. So it's linked with bedding, clothing, and all kinds of fabrics and goods. We also have another type of result here. It's a negative result, but it is very, very interesting also because it shows that the words rat and flea are not used at all. So rats and fleas are not identified as a source of the plague here in Marseille. So what can we say about the fact that fabrics were thought of as a source of infection? Well, if we listen to or read the words of Charles Nobel, who won a Nobel Prize for discovering the role of fleas, we would say that it's the presence of these parasites in those very fabrics that we talked about. And that meant that the fabrics were contagious. If you touch the fabric, you could catch the disease from the fleas. It was also suggested in a study that was printed in 2008 that there are epidemial graphs. The second pandemic, it would seem that the hectoparasites, such as fleas, would convey the plague. And that is something which we have inferred from a historical study carried out. All of the results seem to conclude that transmission is due mostly, there are other causes of course, but mostly it's due to human hectoparasites, given that the most common form in Marseille would be bubonic in nature. Moving on now to the next slide. Here we have an example of someone who has got a problem with a flea, someone who lives in the street, the homeless person. And this kind of picture helps us to understand a little bit more clearly how fleas can convey plague. And in the Middle Ages, people were often infested with fleas and other insects. And you can see the state of this boy's back. Then it's very easy to imagine how these hectoparasites can spark off massive epidemics. Here we have an example of networks of words which we managed to create. They're talking about the sources of the plague. You can see several words on the screen there. All of these words are linked to other words such as infected, fatal, or contagious. And the most recent analysis that we have made was to identify whether there was specific vocabulary linked to plague, and we compared different languages, Italian, French, and English as well. And we can see that there are 29 times more words in common. So that shows that there is a unique lexicon for the plague. So when the doctors talk about plague, they use specific terms, and that vocabulary is worth studying because there is a proper name for each epidemic. There's a common corpus when we're talking about different plagues. So to conclude, this method is a non-biased way of reading the results, and it's reproduced from historical texts. It can be used in all languages, and it's the first time we use negative controls. That's really important to underline. And of course, there are some limits to this quantitative approach because we're talking about the number of times each word appears, and rats and fleas are probably not the most common vectors of plague in Marseille, in Milan. However, bedding, clothing, and goods are probable sources of plague in those two cities. And there is a possible role of human ectoparasites in the purveyance of plague. So it would seem that there is a specific vocabulary dedicated to the plague. So thank you very much for listening. Thank you very much for your presentation. and talking about some of the work which has been carried out, a very important presentation because of the results that you've produced that illustrate new conceptions that one can have about how epidemiology spreads and how plague spreads. We are now talking about the plague in Marseille 300 years ago, but the plague is still rife in certain parts of the world. Didier Raoult said as much. It's highly unlikely that we will have epidemics of plague in Europe. However, plague is still around and some people are still suffering from it. Some even die, particularly in sub-Saharan Africa. I would recommend that there was a plague epidemic in Madagascar just a couple of years ago. So it's a very interesting leitmotif for us to keep in mind as this afternoon goes on. We have taken a look at what happened in Marseille 300 years ago. Perhaps that information can be used to understand what's happening in Madagascar today or in other countries. So that's the very first important subject to take home, or message to take home. So Mr. Barbieri also illustrated what we enjoy doing at our IHU. We like to take what already exists and use it to different ends. So Rémi Barbieri has used concepts which are used in biology to treat experiences in test tubes and on lab benches. He used those to interpret ancient texts which he went to seek out in libraries, texts which date back from the 18th century. So it really is kidnapping in some ways these certain methodologies to use in a new context. So that's very important, a way of working which is completely unique, and we like to foster that kind of approach here at the IHU. It's very fruitful in terms of our intellectual development and our results. So thank you for that first presentation of the afternoon. It's perfectly illustrated how many disciplines are involved in the subject which brings us here this afternoon to understand what went on in Marseille 300 years ago and in other places as well. We need to take a look at anthropology, infectious diseases, many other disciplines which we will be meeting as the afternoon goes on. So knowledge is constructed at the crossroads of different expertises which can come together to shed light on what happened in the past. So I do think it's a very important leitmotif of our meeting here this afternoon. We are happy to welcome today a Scandinavian colleague, Christian Stenset, who is going to illustrate this multidisciplinary approach to play.
This afternoon, I guess you will give your presentation in English. So, welcome to you. Avec une traduction au fur et à mesure. So, Christian?
Thank you very much. Thank you for the invitation. Most of all, I would have liked to be in Marseille now. And it was fantastic to look at Camille, the text. So again, I would have liked to have been in Marseille now. And I'm going to provide you with a procedure. Could you have the next slide? Next slide. Yeah, next again. Yeah, this is what I'm going to talk about. I'm very I'm very happy that we can be here, and I'm very happy also that we can show you can see my some PowerPoint presentation. Because earlier, I have to tell you that earlier I've been at the hospital having an eye operation. I've got my basic. When I told the doctor that I'm going to be speaking in Marseille, he didn't understand anything. You have the next one. This is sort of the typical view of the place. You have the next one. And next. Yeah. This is the view here of the place. This is very much what we see. That is should never ever forget that the plague is a wildlife disease. You have the next slide, please. Next again. This is so. Go back. 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 Did I go back? One back.
Je pense qu'il faudrait revenir à la diapo précédente.
Yeah. This is the view that is common or used to be common from 10 years ago. But again, we should never ever forget that the plague is of mammalian wildlife. So if you're going to look at the ecology of plague, you really have to look at the next slide. Next slide. Yes, very good. So this is really the system that plague is operating, the ecological system plague is operating. And human is only playing a small role here. Mostly wildlife animals are the host of plague, and human is really only a spillover effect from the wildlife. So, can I have the next slide? So, although this is a typical, people is a typical one, you really should look at what is the red circle here, or really the ecological system, the next one. Yeah, this is really the system. If you have to talk about the ecology of the plague, this is really what you have to look at. The next one. Next one. Yeah. And so I will really focus. Can I have the next one? Most of my work has been on studying plague in wildlife systems. Started in Pakistan, then I moved to China, and since then I've been studying this wildlife. More recently, I have also been focusing on human, particularly during the medieval period, and using ancient DNA techniques. And I heard the previous speaker before me was referring to the importance of ectoparasites. I'm convinced that ectoparasites played a key role in the spread of the plague among people. That, by the way, explains also the speed at which the plague moved to Europe when it first was introduced. And also why it disappeared. It disappeared because of increased nitrogen. Can I have the next slide? And press to the next one. This is where it occurs today, typically at the drier places, higher altitude, and burrow-dwelling rodents. So if you put the next one, first go to the next one. Yeah, this is where we find active plague today. You see that it basically is found on all continents, except that in Australia and in Europe. Europe, it has totally disappeared from Europe. But you should never ever forget that plague is a disease of today also. If I could have the next slide. This is where observations of plague cases, human plague cases, is from the period 2010 to 2015. Plague is a disease of today also. To repeat the next slide, we must never ever forget that plague has its main host in non-domestic rodents, which occasionally come in contact with humans. As I alluded to earlier, go to the next one. My focus has been Kazakhstan, and my focus again has been gerbil. Most of my earlier plague work was on looking at or studying plague with gerbil as the main host. But Yersinia pestis can have many rodent species or mammalian species as its host. So go to the next slide, please. You see, it's a very diverse disease with host species, as well as very different vector fleas spreading it between wildlife hosts or spilling over to the human population. I've been fortunate to work with Chinese colleagues in the last few years, last several years actually. Go to the next one. And China is a very interesting country because it is very diverse. It has very diverse plague systems, and we can go very. Yeah. So, and then the next one. So these different host species and different vector species are found in very different ecological systems. So if you ask the question, how will plague respond to climate variation? It depends on where you are. In northern China, there is likely to be more plague cases given the global warming, whereas in southern China, it's going to be less in the face of global warming. We can go to the next one, where actually I started in Kazakhstan by chance, actually. Go to the next one. Go to the next one. They had a very, so this is the world map. You have seen that before. Go to the next one. Yeah. And they had a very good monitoring system in Kazakhstan during the Soviet Union period. Go to the next one. All focusing on this very nice species, the great turtle. This monitoring program in Kazakhstan and the rest of Soviet Union was very important. I'm sure they had some military reasons, but it also had a public health perspective. Go to the next one. Go to the next one. In the past, plague has been a serious problem in the drier area of the previous Soviet Union, in particular in Kazakhstan, as we see here. A few hundred people were infected and died every year. Well, it varied, as you can see, but it was quite a few that was. So it was a public health. Then, next slide. Then the Soviet Union, during the Soviet Union period, the authorities they said that we are going to make a monitoring program, monitoring the plague, and they surveyed the whole wildlife area where plague could be found, and and whenever they found, stop there, whenever they found that there was a local outbreak of plague, they fumigated it. Killed both living using DDT and other ugly things. But it worked. Go to the next. It worked. And yes. So the one to the left is the one you saw before. The one to the right is after this monitoring program was started. And as you can see, very few incidents, the human cases of plague, was observed after this monitoring program. So it works as a public health measure, but it also, if you go to the next slide, it also provides, next slide. But it also provided fantastic data. And this is our very first paper on plague, my very first paper on plague, published in Science. Not because we are good too, but because we have fantastic data. We were able to demonstrate that the great gerbil density had to be over a threshold. If it was below, there would be no local outbreak. Whereas if it was above for the last couple of years, then it might be an outbreak. As you can see by having these dots on the top there, as opposed to lower, only on the lower part, 0. This is actually known from theory, but we, I believe, was the first one to demonstrate this by using real data from a wildlife system. Go to the next one. So this is from Kazakhstan. Now we can move to the next one and the next one again. Yeah, China. As I alluded to earlier on, we have been working in China. China has also fantastic historical data on plague. And it's a very diverse country. Next, yes. These different colors here shows different plague reservoirs. They're basically characterized by different host species. Can we go to the next one? Well, yes, you will go through a sequence now of showing the host species. And if you press several, again, next, you see it's different species. Next, different species in host. That's of course a fantastic situation if you are to study the ecology of plague because of this great variety. Can we go to the next one? Yeah, these are historic observations in China of human plague cases. We asked the question. Can we go to the next one? We asked the question: are there any geographic structure in the temporal dynamics here? And this paper was actually done together with a good French colleague. I have worked a lot with French colleagues. It's a geographic structure, and if you go to the next slide, you can now see in the upper right corner is what we have found a geographic structure in the temporal dynamics of plague, human plague. That compares very, very closely with the different rodent host systems in the lower right. So actually the historic human case data is a good proxy for what is happening out in real world in nature. Can we go to the next? Next. Yeah. So basically, the picture we have in front of us is that you have the dynamics out in wilderness. So whenever the wildlife population breaks down, it has to be above a certain level, with certain density for a couple of years, as I showed you earlier. Then, when the wildlife population crashes, it spills over to other mammalian species, including domestic animals, including people. Can we go to the next slide and the next one? Recently, we have been doing ancient DNA work, but our ancient DNA work, genomic work, is really put into a broader context. We are combining genomics, phylogeny, as well as ecology, as well as history, a professional historian, as well as professional archaeologist. So we really are trying to develop a rather synthetic view. And the picture you get from looking at this multidisciplinary, take a multidisciplinary approach to this, is a very different from the phylogenetic tree. If you only look at genetics, it's very clear that plague stayed in Europe during the medieval period. That is not necessarily the case if you dig in other disciplines. And here is one study we published in PNAS a few years ago, a couple of years ago, where we showed at the beginning of the medieval period, after the Black Death, it was primarily from Russia. You see the two arrows coming in. And this is most likely come together with fur trade. Earlier this year, we have another paper in PNAS taking a little bit more recent period. And the outbreak in Marseille in 1720 is also our case. It is very likely that that came from Turkey, as well as the one in Sweden, most likely come from Turkey. So Turkey and the Caucasus, Georgia and the southern part of Soviet Russia, is likely to be our stores feeding into Europe repeatedly. And this is a picture we get. This is a picture we get later. This is a picture we get from combining different disciplines. If you only look at genetics, you will say that there must have been a reservoir in Europe. Whereas if you bring in other disciplines, not the least ecology, climatology, you will see that there is most likely hypothesis that plague came into Europe several times during the medieval period. With this, I'd like to thank you very, very much for your attention. And once again, I would have much loved to be in Marseille today.
Merci beaucoup, Christian. Well, in fact, you were present with us this afternoon, so thanks so much for your speech. And qui était une présentation également extrêmement intéressante.
Which was an extremely interesting presentation and follows a certain number of running threads of this afternoon. And recalls once again that the plague remains an infectious disease that is still killing people on Earth in a certain number of countries that were clearly illustrated by the different maps presented. And we need a multidisciplinary approach. It is essential in order to understand the plague epidemics. And we must remember that the plague, and it's true for other infectious diseases also, but they're diseases, as Didier Raoult says, that stem from ecosystems. So we need to look beyond the patients that are affected, beyond human populations, and we need to look around us. So for the plague, we need to look at the animal life, and we saw the important role of animal, and beyond animals, there's the role of the inanimate environment. And for the plague, the possibility was mentioned that soil can be one of the virus, and then it is passed on from host to host. Thank you very much for this presentation. So we're going to continue our journey throughout time, and I think that we're going to go quite far back, maybe beyond 1720 in Marseille, with a presentation of Nicolas Strascovan, who we have the pleasure of welcoming this afternoon. I think that Nicolas is in the premises of the Pasteur Institute, where he is working after having worked with us in our laboratories in Marseille. And he initially came from another continent. So Nicolas, thank you for your presentation that you will be giving either in French or in English. We will find out now. Thank you, Michel, and thank you for your invitation. I'm at home. I'm working remotely. And I would like to know if it's possible to put up my presentation. And there's an echo. Thank you. I'm going to try and do the presentation in French. I'm still struggling, I'm afraid, to master French. It's not an easy language to master. The title of this presentation is The Paleogenomics of the Plague. And by paleogenomics, we mean the study of the entire genome of pathogens and data analysis, and I focused on the plague. So there is a long history of pandemics throughout our history, and promoted by virus such as measles, AIDS, and today COVID-19. But there are also some terrible pandemics, and the most mortal of these was the plague. The plague the most famous ones being the Black Plague in the 14th century that killed between 30 and 50% of the European population. But we can also mention the Marseille Plague that killed a large number of inhabitants 300 years ago in Marseille. And as Professor Raoult said, he was in the work done by Didier Raoult. We revealed that it is possible to collect DNA samples of the pathogen from remains, human remains. And this work, which was published in 1998, showed the beginning of the disease in Marseille. So one decade later, we now have technology that allows us to produce a large number of sequences, and we have now identified the entire human genome. And so we can do DNA sequencing far more easily now than with older technology. So as from the introduction of this new technology, we can now collect human DNA from archaeological remains, and we can study the entire genome, and we can piece together pieces of human history that was inaccessible so far. For example, the origin of our species in Africa. And we see how the populations in prehistoric times interacted and migrated throughout the world. What Didier Raoult's presentation showed us that they managed to collect a complete genome of Yersinia pestis from the victims of the Black Death, the human victims of the Black Death. From archaeological remains dating back to the 14th century. And this is what we call paleogenomics. It's the analysis of the entire genome of ancient pathogens. So teeth are very composed of very resistant tissues, and therefore it is easy to collect DNA samples from them, which is why teeth were used. In the plague, the plague had a great impact on human history, and there were a number of plague pandemics, and the most documented one is the first pandemic in the 6th century that killed. It's what we call the Justinian plague, and it killed about 100 million deaths. Then there was the Black Death in the 14th century that killed between 30 and 50% of the European population, and a third pandemic that began in the 19th century and carried on to the 20th century. And as Didier explained, it is unfortunately a plague that is still around, but that can be treated with appropriate methods. So different laboratories around the world have managed to collect the complete genomes that dated back to this pandemic. And there are 93 ancient genomes now that have been obtained dating back to these pandemics. And so what type of analysis can we do with this type of data? One of the analyses that we can do is what we call phylogenetic analysis, which you can see an example here. In the phylogenetic analysis, we managed to identify the family tree of the ancient strains of pathogens. You can see the connections between the different strains And at the top of the tree, between the most ancient strains in this genealogical tree, and the strains at the bottom, which are the most recent strains. So the Great Pest of Marseille has been explored significantly by Didier Raoult, in particular the work published in 2016 by Boss et al. And they identify 5 complete genomes of dating back to the plagues from remains excavated in Marseille.
With analyses which they carried out, we used information of the entire genome to construct this family tree, if you like, to understand how the different strains of plague can be linked up. And that work has shown that the different strains that produced the plague in Marseille was almost the same strain as that which produced the Black Death in Europe several centuries previously. So with these analyses, we can better understand the epidemiology of this illness. But as has already been said in the prior presentation, this biological data has to be correlated with contextual and historical data so that we can better interpret the data. And that is work which we've been carrying out recently. So another interesting discovery was the discovery that plague was already around in the Bronze Age, and that was amazing to discover because we don't really have any documentation about that kind of infection, and that is why approximations were made which have made it possible to identify traces in different zones. So in 2015, one researcher said that that the plague had been in many different places in the Neolithic period. Some work was published 2 years later in 2017, and it illustrates the ancient genomes from that period, beginning of the end of the Neolithic period. The genome had been localized. You can see there on the map, and it has been shown that different strains were in different places, and it was said that the plague came to Europe because of massive migration from the steppes. You can see the arrows there, steppe ancestry. So discoveries were made in Marseille, which were quite unexpected, and that was the presence of the plague in Europe some 5,000 years previously. That's the most ancient case of plague that's been discovered to date. And the biggest conclusion was that the plague that was in Europe before the migration from the steppe did not correspond with the first model that had been developed. It's very interesting with this discovery. At that time, towards the end of the Neolithic period, we found plague all over the place, in very distant places, places separated greatly from each other, and we wanted to understand why. Why was plague scattered all over the place like this in these zones which were very, very separate from each other? Phylogenetic analysis have shown that this particular strain was not only the most ancient in terms of the date, but it is the closest to the common ancestor of all known strains of Yersinia pestis. So all lineages are linked, even those strains that we know today in our modern world. They all have a link back with these ancient strains. So we can say that there was a huge spread of this lineage of plague. So one might wonder, what happened in Eurasia between 6,000 and 5,000 years ago? How can we explain the spread of this plague? So we worked with different colleagues in Sweden, and the Swedes helped us to interpret the results which we had come up with. In that period, there were some new technologies that were introduced into Europe from Eurasia, and there was also animals which were drawing vehicles, and that meant that we had the first trade routes open on a big scale throughout Eurasia. And so that's a perfect condition for the spread of a pathogen over a long distance. And that too was a time where, for the very first time in the history of humanity, in Europe and Eurasia, the very first time, we found those huge agglomerations. In Europe, these were colonies. There were up to 20,000 people living in these conglomerations, and these populations lived in conditions which weren't absolutely optimal, as you can imagine, and that will have fostered the occurrence of pathogens. So there were the perfect conditions there for pathogens to propagate and develop. And they spread on a large scale, thanks to, amongst others, animals which could transport people further. So, for the very first time, the conditions were rife. So, using all of that information, we created a model to illustrate the emergence and the dispersion of plague in the Neolithic period and the Bronze Age. And we can see how far everything spread on those trade routes, thanks to those new technologies that had been invented. It's very interesting also to see that immediately after that event, we can see that the population in Europe shrank. In some areas, people actually disappeared altogether. And that is the period which we call the decline of the Neolithic. And immediately after that, we see a massive movement from the Eurasian steppe, which changed once and for all the genetic foundation of the humans in this area. So thanks to all of this work, we see that plague could have played a fundamental role in the Neolithic decline and in the genetic change in Europe. I'm now going to present to you a new aspect of the studies which I carry out, analyzing the anthropology and the biology of the plague and tying that in with IT. So we can carry out analyses with the life sciences, anthropology, and now we can add IT into the mix to develop analytical tools and computer tools that will help us to clear our data. And I would like to thank the 3 people who are listed on the top left-hand corner there in the Paleolithic study which I carried out. I'd also like to thank Michel Drancourt and Didier Raoult, who have invited me to speak this afternoon, and also to have inspired me to look closer into this thrilling area of paleogenomics. And I would like to thank them for hosting me at the IHU. I'd like to thank the Pasteur Institute also, and the Microbial Paleogenetics Department. And I'd like to thank everyone, and I'd like to thank you out there for listening to me. Thank you very much. Thank you very much, Nicolas, for that wonderful presentation, for bringing further information for us to digest and to help us better understand how plague epidemics can have come to Marseille and spread throughout the zone. And you also showed us a relationship in both directions between populations. On the one hand, we saw in the 2 previous presentations plague that seemed to be following the flow of human beings along trade routes and migratory routes, and on the other hand, the plague that also played a role, because it was a deadly illness, it played a role in modifying the structure of the populations that it infected. So it was very interesting and enlightening to hear all of that. And of course, it's a very hot topic, and these infectious illnesses would follow the populations and set up in a brand new area, according to the speed of populations being moved around. And over recent months, we've been able to see this also with the COVID pandemic. So without further ado, after that charming journey, which we've just been on, a journey through time, which took us back to the Neolithic age, we are going to continue our travel in time as we hear from Stefan Tsotzis, who is going to talk about a multidisciplinary approach to ancient infectious diseases and see what anthropology can contribute, which he practices at the medicine school here in Marseille. So, Stefan, thank you very much for being with us. Well, thank you very much for having me. Indeed. I'm going to start out with a question. It's at the beginning of my presentation. Is there such a thing as anthropology of plague? Well, what a mega question to start with. But this question is quick to answer, if we can just look at the next slide, because it simply means that you have to look through the different tracks to see that plague really is a subject for anthropologists. Anthropology is a holistic science. It has many dimensions: biology, society, culture. And so studying the plague in all its many aspects corresponds to what we do. And it's an open field, it's a vast area, because apart from being a biological fact, plague is also a social phenomenon. And the field is very huge. But I am going to limit my presentation to what interests me mostly. That's anthropology to begin with. The anthropology of death via a certain methodology. Let's move on to the next slide here. I'm going to talk about archaeoanthanatology, death and deaths. Okay, archaeoanthanatology is a subject which is out in the field. It's biology, anthropology, all mixed together. We go to burial sites and study the remains of beings and all that can be found in the soil and which interacts with those human remains. So, what's our doorway into such an event? Well, it's the question of how funerals were managed and when they reached an abnormal scale. So above and beyond calling into question their usual way of life and their social practices, because of the speed and the intensity of demographic function, then civilizations had to find the right solution so that they could continue to survive in a particular context. And of course, they had to manage many more deaths than in the past. So they had to adapt to this new situation. So that leads to quite a few problems for the specialists that we are. Problems which can be summed up as what in those periods, what were funeral practices, what was conventional, what was usual, what was normal, and what was abnormal in those days. What kind of archaeological clues can we come up with for those different practices? Those unusual practices. Well, first of all, it's where were people buried? Where were the bodies put into the earth? What kind of processing was used? And if there was some special kind of treatment involved, did it become the norm in a specific context? So the source of our specialization, well, I've already talked a little, well, you heard a little bit about it in the previous presentation. So these are the different sites which can be discovered, and then digs can be carried out and have been over the past 30 years. So we can dive back to ancient times, to proto-history times, and so there's a lot of information linked to pandemics, particularly to the second pandemic in the Middle Age, the Black Death, which we've already spoken about today, and then moving into the modern period. A lot of these sites are in this time frame, those sites that have been analyzed so far. Earlier on, I was talking about the question of the places where the bodies were buried. So what do we see when we look back in time for the second pandemic? Well, there's a lot of documentation. Archaeologists have done their work. But paradoxically, for this Black Death, they were mainly the traditional burying places that were used, the traditional cemeteries in each parish, and the burial rites were pretty much as the norm. They had individual tombs. There are examples of specific places dedicated, or rather, there are very few examples of special burying places just for plague victims. So that was an interesting thing to find. However, depending on how intense the epidemic is, there are some practices which are quite alternative without changing the place where the bodies are buried. So there are graves with many bodies in, several bodies in the same container and at the same time. So that is slightly different to the norm. And all these adaptations are made. However, we know that during the Black Death, people wanted to maintain as much as possible their traditions, particularly in terms of the way the bodies were positioned, lying on their back, which way they were heading, from east to west, and also the way their legs and arms were folded or straightened. So as of modern times, as of the 16th century, things started to change a little bit. And we discovered specific zones where plague victims were buried. These zones were often linked to the different structures which would take care of the ill, the infirmaries, hospitals, etc. And during this period, we see specific places being created for plague victims. There are some individual tombs, however, there are also mass graves.
So, in addition to having specific locations, whilst continuing to have mass graves, there's also the fact that funeral rites changed in the position in which the bodies were placed in these mass graves. So there are different positions, bodies head to foot, and things that are not at all align in terms of the posture of the legs and arms. If we zoom into our region, which has certainly been the subject of many studies, because we have 7 sites of excavation of burial sites, we can see these different types of cases. If we go right back to the 16th century, which is the beginning of the modern period, and here we see a cemetery in Fedon, which is in the Bouches-du-Rhône, and we can see that this is in a parish cemetery near an infirmary, and we have individual graves, but from time to time we also have mass graves with a variable number of individuals. Similarly, on this site, in Puy-Saint-Pierre in the Hautes-Alpes, so it's a cemetery just outside a village. It dates back to the 17th century, so it's the Epidemics 1628-1630. And we can see that although the grave is well defined in terms of surface area, there are different configurations with individual graves in standard positions and mass graves with a chronology that we could restitute in the side. So to the north we have the individual graves, and in the south mass graves, which seems to indicate that the closer you get to the exit of the cemetery, there was an increase in the number of deaths that had to be managed urgently, and which meant that the funeral rites of that time were transgressed. So what's the best documented in our region is the plague of 1720 to 1722 in Marseille and in Provence as a whole. And there are a certain number of reference sites within the city of Marseille and the city of Martigues. In the city of Marseille, when we look at the different archives that are available, as we were able to do, we know that a certain number of mass graves were created in the city, and 2 of these graves were found and excavated by archaeologists. So there's one beside the Major Cathedral and the other on the site of the observance. And so the first corresponds to a mass grave of 1720, and the other in a second wave in 1722. So in Marseille, near the Major Esplanade, what was excavated was part of the grave, not the entire one, because it was covered by later structures. And you can see this is nothing like what we've seen, organized cemeteries, even though there were mass graves. We're really seeing here disaster graves with lots of individuals, one on top of the other, with no apparent organization, and all types of uncustomary positions. But nevertheless, there are some individuals that seem to have a more conventional position, as if they had been placed intentionally in this manner, in particular at the bottom of the grave. So which also illustrates the urgent situation. It's when we come back to the chronology of this funerary structure, and that's the fact that there are archaeological artifacts that were found, in particular remains of clothing or small objects. Or we can see a shoe buckle and fragments of leather, which demonstrate the presence of a shoe. And what you see at the bottom are everyday objects, in particular coins. And these coins are very important because they're silver coins that had a certain value. So it shows that the bodies were not shown. Also, we have on the site of the observance, we see the same thing. And this is what we can see on the next slide. There is some anticipation of deaths because a very large grave was dug and a far larger space was developed for mass burials. Of course, this is a second wave, and a number of deaths that were far greater had been anticipated. And in fact, there were fewer deaths than anticipated. So in Martigues, there are also 2 sites that were excavated. Once again, this is a disaster grave, but the organization is not the same. They are trenches that were dug parallel to each other. And there are a lot of elements that are common to Marseille. We can see people are buried. There's the use of lime. And there are individuals that have not been searched for everyday objects. And the bodies have been placed in a variable manner because it's not necessarily the same between the different trenches. Here you have some examples. And this is the example of bodies placed, poured out of the carts directly, and with some separations between these groups of skeletons. So if we summarize things, there are over time, in terms of practices and the treatment of bodies, there are a certain number of variations. There are variations that are very clear, mass changes from the modern period, where we no longer have usual practices because we prioritize health issues. And you understood that from this data, by putting things in the context of funerary sites, we can see that these funerary practices give us a lot of information that indicate the fact that there was a plague, although it's not sufficient, because even in the framework of a mass grave, this could be the result of different types of catastrophe. So other elements must be taken into account, of course. And first and foremost, this is what we call funerary recruitment in our jargon. When we see the example of Martigues, What we noted first from the parish registers, the historical archives, is that even though there was a strong reduction in population, the plague did not modify the structure according to age class. So this is shown by pre-statistical archives. And when we compare this data with the archaeotechnological data, and we look at funerary recruitment of structures related to the plague, we can see that the structure of the living population before and after the epidemic is the same as the composition of the sample of skeletons that are found within these burial sites for plague victims in Martigues. So we have a picture of the population before and after the epidemic. And this specificity is what allows us, under certain conditions, within the context of an epidemic, to say that this is directly related to the plague. So, funerary practices are very important elements, but not sufficient. Funerary recruitment is once again a very important element, but once again, it needs to be added to, and we need to add to this with what will be the object of the next presentation, the possibility from this osteoarchaeological set to highlight the contamination by the Yersinia pestis bacilla that are found from osteoarchaeological remains. But that is the domain of immunodetection and molecular paleomicrobiology, and that's another story. Thank you very much.
Merci beaucoup, Stéphane, pour cette présentation.
Thank you very much, Stéphane, for this extremely rich presentation, which does indeed show that many of these studies were carried out here. So what you're doing, what we're doing, is based on history. And we've understood that the history of the plague is well explored in Marseille, as it was an entry door to the disease, as we can see from the routes presented in the previous presentation. So I'm going to actually carry on from your presentation to do mine, because we've reached the points where anthropologists have revealed skeletons on different occasions that are buried, for example, at the site of the Major Cathedral in Marseille. And this is how the history of paleomicrobiology came, and that they turned to microbiologists, and I'm one of these, and asked us, well, do you have the capacity in your microbiology labs to diagnose the plague, to demonstrate that among these human remains, the microbe that we've been talking about since this morning, Yersinia pestis, which is the bacteria that causes this mortal affection, the plague. And so that's what I'm going to present now. It's how in the microbiology lab, which is currently in the IHU Mediterranean infection, how we can confirm an infection by Yersinia pestis. So we saw in previous presentations that we do have historical data. And for once, I haven't shown a painting by Michel Serres that can be admired in the Fine Arts Museum of Marseille. But this picture is in the Marine Museum in Paris, but it also shows the port of Marseille. This port, which opens out onto the Mediterranean to the east, and that is where the plague came to us in 1720 with the ship, the Saint-Antoine, and with a whole series of events that Didier Raoult presented in the introduction of this conference, where we can clearly see that financial interests provoked the disaster, which was this very high mortality caused by the Great Plague in Marseille. So we have historical data. This is a painting, Remy Bardinet, you showed earlier on, that we also have texts that recount this event. Next slide. We can see also an iconography which shows the extension of the plague, in the spread of the plague in Marseille. You have the structure a few years before the Great Pest of 1720. This is what the city looked like. In red, you have the old quarters, the panier, as we call it. And on the other side of the port, what at that point was the new part of the city, around the arsenal for the great ships. And 1720, Louis XIV built this with the cannons facing the panier neighborhood rather than being directed towards the English who were waiting outside the port. The next slide illustrates the protective structure that was developed. On the left-hand side of the slide, you can see a building that we're all familiar with. You can still see it. We all go past it when we go walking towards the Musen Museum, for example. And this building, rather ironically, dates back to 1719, the year just before the Great Plague of Marseille. And it housed the health officers, who were municipal officers who were appointed to embark upon the ships waiting at sea, just outside of the port of Marseille, to check that these ships were not carrying any plague. So it is this system that, for a long time, protected the city, but once again, failed during the summer of 1720, which gave rise to the massive disaster that ensued. So here we see the plague that actually came in through the commercial port called the Joliette, which is up at the top right-hand side of this slide. Along the coast, you can see the first ramifications with the black arrows, which present the plague spreading through the city. We can see that this began in the Joliette neighborhood, and first of all spread throughout the old city of Marseille in the Panier neighborhood, and then they went beyond to invade the new city, which is in orange on this slide. And so it is the entire city of Marseille, ancient and the modern city of the time, which suffered from the plague in 1720. And as presented, this then spread beyond the city of Marseille. And in just a few months, the plague became a regional problem, with measures, preventive measures that recall exactly what is happening now. This was in 1720. The King Louis XIV has died, and so the King built what we call the plague wall. And when you go walking towards the Luberon region, we can see this plague wall, which actually was intended to circle Lower Provence, where the plague was rife, and this was intended to restrict the movement of population. We can't see it on this slide, of course, but there were pomegranate trees that were planted, and there were soldiers standing along the wall.
Du Tour à ce moment-là, eh bien, était de savoir si nous avions une capacité.
In our microbiology lab, the idea was, could we detect whether these skeletons that were unearthed, as Stefan Just presented, to know whether they were carrying the Yersinia pestis bacteria. And so we were able, and I'm thinking that this is, for the very first time in the world, we showed that it was possible to detect traces of molecules, so a fraction of the chromosome of the genetic memory of the microbe, and therefore human remains. And this is something that was invented by Professor Raoult, and the idea was to use the tooth and the pulp within the tooth. So it's in a dental pulp that we work on in order to trace ancient bacteria. So we must underline the contribution of Dr. Gérard Dulem and the odontology faculty of Marseille, who developed the technique, the initial technique that's today allows us to trace these microbes using ancient dental pulp. Next slide. This is the first work as they were published, as they were shown in previous presentations, and which illustrate this. And as Didier Raoult said, we first of all had to work on ancient dental pulp, and we invented a working methodology, a way of tracing the DNA fragments of the microbe that we call a PCR suicide, which allow us to avoid all risks of contamination and all risks of what we call false positive, to make sure that we're not mistaken when we say that this skeleton does contain the Yersinia pestis virus. This is what we achieved gradually in recent years. And this is that we've shown that, as Nicolas Strauss-Govan presented, this dental pump is a highly vascularized organ. And beyond the dense vascularization, what we understand now is that each dental pulp within an ancient tooth contains a minute drop of blood that remains in exactly the same state as it was upon the death of a person. And so you see on the lower part of the screen on the right of these photographs, you can see what is called modern blood, and that is a sample, a modern day sample of blood. So it's just as if I had just had a pinprick on my finger, and we look at the drop of blood with a microscope. This is what we see. And all of the rest of this slide are ancient dental pulps. And the idea that I'm trying to present here is that the red globules that we can see on these pictures, whether they're red globules, blood cells from the 19th century or the 16th century, or in the middle we can see on the bottom the red blood cells from the Roman period. You can see from a site in Besançon in France, the site of Vioc, which dates back to the 1st, 2nd century AD. All of these have red blood cells. So within this ancient dental pump, there's a small drop of blood that is perfectly conserved, and we use this in order to trace the microbes such as that of the plague.
Une autre illustration de ce que je viens de vous présenter.
Here we have another illustration of what I've just been talking about. I'm no longer talking about the red blood cells, but the white blood cells in our blood. We have white blood cells, and these white blood cells help destroy microbes. The white cells destroy the viruses that are responsible for COVID and all kinds of microbes, including plague. And here we have white blood cells taken from a person who died during the Roman period in the 1st century after, in our time, AD. And so we can look at those white cells. I could do the same thing and prick the tip of my finger and I would see similar white cells. So we realized that this dental pulp contains blood that is perfectly conserved. Imagine discovering white blood cells that are 2,000 years old, and they are perfectly identical to the white blood cells that you might take from someone today. Here, using that knowledge which we managed to glean of that tiny drop of blood which is trapped in the dental pulp, above and beyond all of the other techniques which we've discovered over the afternoon, which aim to detect DNA, i.e. the genetical memory we managed in our laboratory to develop, or in Didier Raoult's laboratory, we have managed to trace other typical molecules, they're proteins typical of the microbe. We work a lot by opening up a new field. We call it paleoproteomics. And this new discipline enables us, on the one hand, to trace the microbe, the Yersinia pestis, throughout the Marseille plague, and we have results here which were obtained from the Delos site, which Stefan talked about. And on the next slide, you will see how we can interpret those results to fuel a new field of specialization, which we've recently invented with Didier Raoult and other students, called paleoseriology. And in that single drop of blood, there is also serum, as we have in our blood today. And in the serum, as you probably know, in the serum there are those antibodies. So antibodies that we are tracking in the laboratory to try and track down the elements that are responsible for COVID. Those antibodies can be detected in ancient samples, and you have an example here. It's a bit of a technical illustration. Apologies for that. But every single black line which you can see on those red strips, well, each black mark corresponds to an antibody that has been detected, an antibody which has been taken from a drop of dental pulp, drop of blood in a dental pulp, which dates back to the 17th century. So here we are setting up new lab techniques, and constantly in our lab and in others throughout Europe, we are constantly putting in place new techniques which enable us quite modestly to carry out diagnoses, to identify which microbe infected a specific individual when that person died. So we carry out a diagnosis, a retrospective diagnosis of an infection. So what does that give us? Well, it means that we can diagnose ancient illnesses on skeletons from the cathedral site here in Marseille. It enables us to better understand the epidemics of the past, to better understand the different options, the different epidemiological chains which can be set up during an epidemic. And understanding what happened in the past will enable us to understand better what's happening today. Let's take the example of plague. As was presented earlier on, we've understood that it is an illness that is not only an illness in mankind, but more so, I would say, an illness in animals that surround us. Christiane Stenset gave us information on that front. So plague came from animals. It was from an illness which man caught from an animal. And this paleomicrobiology work has taught us that at the beginning of this epidemiological chain of plague, there's animals, and before that there is the ground, and that's something we continue to work on in the lab. We have finally understood more precisely the fact that this Yersinia pestis is a strain that can survive in certain conditions in the ground. It's illustrated here where you can see a map of North Africa, where you can see Algeria. As we heard this morning from one of the speakers, and Didier Raoult echoed that when he was talking about Oran, and the plague, the book by Camus. So there are outbursts of plague in Algeria from time to time. And what we have understood is that these resurgences are probably linked to the fact that this microbe is quite capable of surviving in the ground in certain conditions, conditions that we now understand. It's all been presented very clearly by Christiane Stensen. These dry conditions, and we've understood more recently, thanks to Rémi Barbieri, the ground has to be arid, dry, and slightly salty. There has to be a bit of salt in there in the ground. And that possibly explains, in part, why there's no more plague in Europe right now, because right now that specific condition of an arid, salty ground is just not to be found in Europe. Whereas just a stone's throw away, just across the Mediterranean which brings us all here together today, in Algeria, that that collection of characteristics are present. So this knowledge had already been provided by Alexander Yersin, who discovered the microbe. When we read his write-up of his discovery, Alex Yersin says that he was able to cultivate this microbe, the microbe which is responsible for plague. He could cultivate it by taking samples of ground, of soil, from the houses in a neighborhood in China where the illness had been found. So let's be honest, we rediscovered some knowledge which had been slightly forgotten over the decades, and we've modernized it and we fine-tuned it. And so this is a first link, probably the first link, the very important first step in the chain of events which lead to the human plague. And a second example on this next slide. Here we have an extension of that work carried out by Barbieri on the ground. Some minute work was carried out on analyzing the aridity and the salt levels in the soil in the United States. This is something which was published at the beginning of 2020. And on the bottom left-hand picture, you will see that plague, human plague in the States, because the United States are one of those countries that declares plague levels every year to the WHO. So those dark red points are where we have cases of pest which correspond to the yellow parts in the map, where the ground is arid, and the blue parts on the top right-hand map where it shows areas in the United States where the ground is salty. So for the time being, we have this vision of the fact that at least in the Northern Hemisphere, there are regions in the world, in the Northern Hemisphere, where the salinity and aridity of the soil will foster the survival of this microbe, which leads to plague. This slide illustrates some more important information which we have gathered thanks to our work. This work has already been referred to today. We have these human ectoparasites, and they have a flea here on the right, a body flea. You can see it's slightly kind of fluorescent there. Well, this flea, which Rémi Barbier mentioned, was a vector of typhus rather. Well, this flea is also a human ectoparasite, which is an agent for conveying plague. So the floor is really opening up, as Didier Raoult has said. Some people have experienced this. We talked about rats and fleas on rats, which would give the plague. But now we're opening up the horizons, really. It's not only the fleas which are on rats which will pass on plague. If we take the Marseille plague, for example, there were also these human body fleas that could be a vector. These fleas were not only on the bodies of mankind, but they were also in the clothes that people would wear. And when you look for bodily fleas, you have to look in the seams of undergarments to identify them and to find them. So this is, it may seem like a detail, but it is so important to understand that at the very foundations, the infestation of these bodily fleas, which are a vector of plague, well, that infestation is linked to clothing and to hygiene of the clothes that you wear. So when you're fighting plague, you have to be careful about what people are wearing, not only keeping their bodies clean, but they have to keep their clothes clean. So you soap your body, but you also have to make sure that the clothes that you're wearing are perfectly clean also. They have to be free of these ectoparasites, which are vectors of terrible, fatal illnesses, such as typhus and plague. So this paleomicrobiology work, what we're carrying out in Didier Raoult's laboratories, and which has been carried out elsewhere in Europe, well, this work leads us to work on ancient illnesses in Marseille and elsewhere. And it explains to us, it allows us to understand how the plague will unfurl, and we can identify different scenarios, and we can learn from that past experience to tackle current hubs, so current clusters. So it's important to identify these ectoparasites, these tiny animals, I could call them, these minuscule beings which live on humans, and they are clusters and triggers of these illnesses. So to conclude, the work that we carry out aims to renew and set up lab tools that can offer a diagnosis of plague, but also a diagnosis of other illnesses, which are always responsible for epidemics. And this work has enabled us to show that the tooth and dental pulp are so important for working on these ancient epidemics. Everyone who works on ancient microbes is working on this invention, which we discovered here with Didier Raoult some time ago. I'm not going to say how many years ago. And I want to insist on the importance of including several disciplines when you want to understand the dynamics of an ancient epidemic such as that plague here in Marseille 300 years ago. It is so important to work hand in hand with historians, anthropologists, and with people who are familiar with the ecology, those who know the ecology of animals, and perhaps also we need to work with people who are familiar with the ecology of the ground. So my presentation sums up in some ways the previous presentations that we'd heard this afternoon, and I think I can now wrap up our meeting by thanking first and foremost the participants here who have weathered these difficulties that we're faced with thanks to COVID. So thank you for weathering the storm and being with us here. Thank you for your determination to share your time with us this afternoon and present the wonderful work which you've carried out. You're putting in so much to our knowledge about this medical field. I'd like to thank the Regional Council here for the Sud Department who's hosted us here. I'd like to thank all of the people who behind the scenes are making sure that everything runs smooth, technically speaking. And I think, having expressed my thanks, I think now the time has come for me to close this seminar. And how ironic it is that they're celebrating the 300th anniversary of the plague here in Marseille with the COVID-19 epidemic also. How ironic is that? So thank you very much for being with us.